Good morning, everyone, and welcome to your favorite science show, Lab Time. Let me introduce you to the subject of the day, patch clamp techniques. You may be wondering, why make a show about it? Let me answer you with an example. As you probably know, calcium has a significant role in many cell functions. It is, for instance, involved in cell proliferation and apoptosis regulation, which are fundamental mechanisms in cancer. Calcium, like many other ions, goes in and out of the cell through channels. The study of the fluxes that go through those channels gives us many pieces of information. And this is why patch clamp is so important. Patch clamp was developed in the late 70s by Erwin Neher and Bart Sackman in Göttingen. They received the Nobel Prize of Physiology and Medicine in 1991 for being the first able to record a single channel activity. We will, in this episode, have a quick tour of the material needed to realize a measurement thanks to patch clamp. Then explain the differences between two configurations, whole cell and on-nucleus. Finally, we'll conclude on the usefulness of these techniques in research to answer the following question: why can we say that the development of patch clamp techniques had a significant impact on physiological studies? But I've talked enough. Let's leave it to experts.
Before the patch clamp technique was developed by Neher and Sackman, scientists used the voltage clamp configuration. It's used to record the ion currents through the membrane of excitable cells, and it's done by controlling the transmembrane voltage by injecting current inside the membrane. The difference between patch clamp and voltage clamp technique is that the first one allows scientists to see the activity of individual molecular ion channels, while voltage clamp is used to study the whole membrane. So, although both configurations allow to draw conclusions about the conductance, patch clamp made it possible at a smaller scale. Now that we talked about the origin of patch clamp, we can see what the technique really is. So patch clamp involves a glass micropipette forming a tight gigaohm seal with the cell membrane. The micropipette contains a wire bathed in an electrolytic solution to conduct ions. To measure single ion channels, a patch of membrane is pulled away from the cell after forming a gigaohm seal. If a single ion channel is within the patch, currents can be measured. There are 4 different configurations of patch clamp based on the attachment of the micropipette to the cell. So we have cell attached, whole cell, inside out, and outside out.
Thanks, Professor Ferrante. Now, let's see what a lab in which we use patch clamps looks like.
Introducing patch clamp setup. We will introduce to you the different tools and machines used in a patch clamp setup. The first is amplifier and data acquisition. Amplifiers measure changes in membrane potential or current resulting from influx through ion channels. The data acquisition converts analog signals into digital signals. The second is the microscope. Microscopes are optical tools to scale up the samples. Different types can be used for different projects, like for slices, fluorescence, optogenetics, and many more. The third is perfusion chambers. The perfusion system allows a fast and complete exchange of the solutions used during the patch clamp. The fourth is pump or the vacuum. The pump will complete the perfusion and add a temperature control system. The fifth is the micro manipulator. Patch clamp manipulators are highly precise instruments. They move the patch pipette with nanometer precisions. The sixth is Faraday's cage. The cage around the patch clamp rig shields noise from interference sources in order to get a perfect signal-to-noise ratio. The seventh is the table. An anti-vibration table is required to compensate for vibrations which can disturb the recordings, because even small vibrations in the picometer range can break down the seal. Eighth is the PC and software. A powerful software to control the amplifier, record and analyze data is a critical tool to produce solid results. The ninth is temperature control, to run the experiment at a constant temperature or to perform a temperature-dependent recording. The 10th is stimulus generator. Brief pulses of electricity are used in the research application as stimulus to excite nerve or muscle fibers. The 11th is the puller to make the pipette from the glass or quartz. The 12th is the capillary, cover slips, and other accessories which are required for patch clamp.
Thanks a lot for this clear presentation of the setup. Now that we know what is used to realize the measurements, let's see what they are used for with the first example of the whole cell configuration.
Let's talk more specifically about the type of configuration. The technique of pad clamp whole cell configuration is more common for measurement of ion currents and the only one enabling measurement of action potential. This method allows detailed analysis of mechanisms and structural determinants of voltage-dependent gapping of ion channels, as well as regulation of channel activity by intracellular signaling pathways. For example, we have the experiment from the Journal of Neuroscience Methods, Volume 59, Issue 1, which illustrates how application of the whole cell technique to neurogenetic neurons in the rat locus coeruleus in brain slices has led to the finding that cRMP, via its fossilization pathways, modulates tonic pacemaking in these neurons. In the study to be described, the particular advantage of the whole cell technique was that it allowed introduction of the macromolecules related to the cRMP pathways directly into cells. The macromolecules are, for example, protein kinase inhibitor and protein kinase A. This method optimizes measurement of electric events from small cells. The principle of the method is near-perfect electrical isolation of a small patch of cell membrane inside the tip of glass electrode, which is reached by gentle pressing of the pipette against the cell followed by gentle suction applied through the pipette. Electric resistance of the cell pipette junction must exceed 1 gigasil. Under this condition, the background leak currents caused by a flow of random ions between the cell surface and the surface of the pipette tip should not exceed 10 Pa so that the measurement of the current carried through the expressed ion channels is not disturbed.
Thank you. Finally, our last expert will present you a less known technique, the unnucleus, a single channel technique.
While the whole cell technique is used in a lot of cases to give us information about the global activity of the soul, this is not a patch clamp technique that allows the scientist to gather information on a specific channel or area of the soul. Among the many other techniques that give us information on a single channel, the on-nucleus patch clamp technique is a technique that allows us to register the activity of a single nuclear channel. Just like every nuclear patch clamp technique, it requires more precise material and is harder to execute than the whole cell technique, with a lower success rate. In order to perform this technique, we need to fill the experimental chamber with an appropriate bath solution, a solution containing nucleus. Then, after finding and isolating nucleus with the microscope, we can bring the micropipette close to the nucleus membrane and apply pressure from 4 to 10 mm of mercury to create an impermeable seal between the pipette and the membrane. At this point, we are ready to measure the channel's activity. On-nucleus patch clamp is mostly used for inositol 1,4,5-trisphosphate receptors and ryanodine receptors. For example, it was used in the article The Type 2 Inositol 1,4,5-trisphosphate receptor Emerging Functions for an Intriguing Ca2 plus Fluid Channel to understand the mechanism of ATP regulation of the Type 2 inosine trisphosphate receptor. Thanks to the on-nucleus patch clamp analysis of rat and xenopus cells, the authors learned that IP3R2's regulation by ATP is different compared to the other types. Indeed, while the Ca2 plus sensitivity is usually changed by the presence of ATP, it is not true for the type 2 receptor. Furthermore, it has been observed that the IP3R2 displays a more bursting activity in presence of ATP.
Thanks, Professor. And now I'll let our holographic friend, Salah, conclude.
So, as for the conclusion, the patch clamp system is said to be a gold standard in ion channel research. Its different types of techniques have been engaged by several laboratories to identify the ion channel characteristics of a variety of cells. These methods have often been used to analyze in depth the pharmacological properties of ion channel modulators, which are used to treat different kinds of diseases such as asthma, hypertension, epilepsy, and pain. Although the patch clamp techniques can provide a high amount of information, its high labor requirements, a complicated laboratory protocol, and a low screen throughput bring some limitations to their usage. However, the research on the physiological properties of ion channels is a rapidly evolving area of study. There have been developments on, for example, several automated patch clamp systems in the past years, so these limitations are already on the way of being overcome. This fairly new kind of technology changed drastically the study of cellular physiology by offering a high-resolution technique to observe the properties of ionic channels through their activity. Since the discovery of these techniques, the understanding of the role of these channels in important cellular processes, including nerve activity and action potentials, has improved. So that's it. Thank you for tuning in to our studio today, and goodbye and have a nice day.
Goodbye everyone, and see you tomorrow for another episode of your favorite science show, Lab Time.